Welcome back to BinaryBio. If you're interested in bioinformatics and problem solving, you're in the right place. This is the Rosalind Problem Series, where we master computational biology through structured coding challenges. Have you ever played the game Telephone, where a message gets distorted as it's passed along? DNA replication can be a bit like that. When we have multiple related DNA sequences, they're often similar but not identical. How can we piece together the most likely original sequence from all these variations? Today, we'll solve that exact problem by building a consensus string. The biological problem is common. Imagine we've sequenced the same gene from several individuals, or we're comparing a gene across closely related species. We get a collection of DNA strings of the same length. Our goal is twofold. First, we want to create a profile matrix. This is a simple table that summarizes the nucleotide counts at every single position. It has four rows, one for each nucleotide A, C, G, and T, and its columns correspond to the positions in our DNA strings. The value at profile AJ is just the number of times A appeared in the jth position across all our sequences. Second, from this profile, we derive the consensus string. The consensus string is the most representative sequence. For each position, we look at the corresponding column in our profile matrix and pick the nucleotide that appeared most often. That becomes the character at that position in our consensus string. So, how do we build this? Our algorithm can be broken down into four simple steps. Parse the data. Our input is in FASTA format, so we first need to read the file and extract only the DNA sequences, storing them in a list. Initialize the profile matrix. We need a place to store our counts. A dictionary where keys are A, C, G, T, and the values are lists of zeros works perfectly. Populate the profile. We iterate through each position of the DNA sequences. For each position, we loop through all our sequences and increment the appropriate counter in our matrix. Build the consensus string. We iterate through the columns one last time, find which nucleotide has the highest count, and append it to our result string. Our code works perfectly for the Rosalind challenge. But if you're moving into real world bioinformatics, there are three hidden problems you need to know how to handle. First, the tiebreaker. What if adenine and cytosine both appear 50 times in the same column? In our current Python code, Max will simply grab the first one it sees. In a lab, this usually indicates a SNP, single nucleotide polymorphism. To handle this like a pro, you could modify your script to return an IUPAC ambiguity code, like the letter M for A or C, instead of just guessing. It keeps your data honest. Second, missing data. Not every sequencing run is perfect. Often, you'll see the letter N in a sequence, which stands for unknown. If you run an N through a strict ACGT profile matrix, your code might skip it or crash. A robust script should include an other category or a filter that ensures these unknown gaps don't artificially lower the frequency of your known bases. It's all about cleaning your data before you count it. Finally, unequal string lengths. The Rosalind problem assumes every DNA string is the same length, but in reality, deletions and insertions happen constantly. If you try to index a short string using the length of a long one, Python will throw an index error. The fix: use itertools.zip underscore longest. This allows you to pad the shorter sequences with a placeholder, ensuring your matrix remains rectangular and your script stays crash proof. Let's talk efficiency. Let k be the number of strings and n be the length of each string. Both parsing the input and populating the matrix take O of k times n time. Building the consensus string takes O of n. Therefore, the overall complexity is O of k times n, which is linear relative to the input size. Space complexity. Storing the sequences takes O of k times n, while the profile matrix and consensus string take O of n. The dominant term is O of k times n. To recap, we transform DNA strings into a quantitative profile matrix to determine the most common consensus sequence. This is a foundational technique in computational biology. If this walkthrough helped you, hit that like button and subscribe to BinaryBio for more Rosalind solutions. Drop any questions in the comments below. See you in the next video.